start with, okay, I'm going to start with this map of North America. Here's Princeton in British Columbia. And this is the Princeton Basin. And it's the locality that's my PhD locality. Sorry, that doesn't make sense, does it? The locality that I worked with for my PhD is from a site just south of Allenby on the river called the Princeton Church. And it's early to middle Eocene. So it's a weird place. The basin has a lot of coal mining historically, not great coal. And so these dark layers are coal. And if you look at these orange to white layers, that's the stuff where the plants are. It's good stuff. So we have three-dimensionally preserved plants and actually some turtles and some other stuff. At the top of this outcrop, we have some shale with fish. And everything lines up with Middle Eocene. And Dr. Ruth Stockey has been working on this since before I was born. Anyway, so I'm going to start off by talking about anatomical links. So plants aren't like turtles. And great talk, Derek. Thank you. But the way plants grow is both a burden and a boon. We get to have a plethora of information. So plants tend to abort things as they develop. So you can kind of imagine immature fruits or leaves that just weren't going to make it, and the plant just cuts them off and they drop. And so when you have a fossil plant locality, you can use these kinds of little tricks like these anatomical links to put together a plant. And I'm going to talk to you today about Lauraceae. So it's a family of plants. The one that's closest to us is spicebush. And so ecologically, it actually does make sense. Spicebush lives in swampy places. Anyway, I'll go on. But anyway, so we have all the organs that I'm going to talk about that have been probably pruned off naturally by abortion over time. And then preserved. They share these traits, for example, these three main traits. They have these weird cells. We call them idioblasts. Idioblast means weird cell or different cell. And these ones have oil in them or mucilage in modern plants. And actually, if we put a UV light on them, they glow. So that's kind of pretty. And then this is stone cells or sclerids. It means it's a woody cell. It's when you crunch a pear, when you feel that crunch, that's what you're crunching. And then we have these long trichomes, or that's a fancy way in plant biology to say it's a plant hair. So these long hair-like cells. Okay. So I'll start with the vegetative stuff. So we have a plethora. So I think we have on the order of like 40 to 60 floral based organs, which includes inflorescences or isolated flowers, something like 30-ish developing fruits that I've already published in 2009. And these are just the ones that I focused on. There's more. But you can see here, we have these stone cells in the pith. You see them here, and then you see them here. And we have this weird looking structure here. I'll get to in a minute. And then this is a transverse section through a leaf. And this is all very like classic Lauraceae. We have these cells that have autofluorescent mucilage or oil. And then this is really fun. So when you have a bud, so this is actually an indicator of seasonal ecology. So plants are really good at giving us little clues to ecology. We know that the Princeton Church was kind of like a wet place, like a swamp, or maybe like, I always kind of think of it as a bayou. The better we get to know plants, the more I think, oh, we have taxodiaceae, sorry, I'm nervous, taxodiaceae, which is like the dawn redwood, or what do we call that, Ruth? Plants in the bayous, taxodium.
Taxodium.
Thank you. Anyway, and then we, like Selena showed that lovely reconstruction of Sororis. And if you go to the southeastern US, like, northern Florida, southern Georgia, and the swamps and the bayous, you see all these plants together, taxodiaceous plants, Soraraceae. And so in my abstract, I talked about the spice bush because the spice bush is an interesting genus, Lindera benzoin, or spicewood. It includes the variation in that one species, includes all the variation that we see here. But let's take a look here. So we have two big bracts that are enclosing the bud. So you can see it over here. It's on the side of an axis or a stem. And this is actually all by itself an indicator of seasonality. So already we know that the Okanagan Highlands, or that's what the paleobotanists like to call like Maccabee, Horsefly, Princeton, things like that. It's like it was warm, but it was up, right? So it's a weird place. But just from these buds, we can see things like this. Look, 1, 2, 3. We have a spiral phyllotaxy. And this is very standard for the family. And I have never counted all the leaves we have. There's so many, it's not worth counting. So we have, by chance, when we do our peels, these paradermal, we call that paradermal. It means kind of sectioning along the plane of the flat part. And so we can do things like vein density analysis with these plants, which is also used for ecological analysis. But when you look close, you can see, it's hard to tell. It took me a little while to figure this out, but you can kind of see these little C shapes. And then to the side, we have one cell each. And this is what we call parasitic stomata in paleobotany, or just botany. And it doesn't make sense to me at first because what happens in Lauraceae is that it's also a very standard classic type of stomatal orientation. They're actually like little hamburgers. Those two guard cells that are right here in the center, they're actually in the middle. And so they're actually sunk down. So when we look here, you can see one here. You see that? And then the two side by side guys. And then one here, two little guard cells, and then side by side. And these are the epidermal cells. And normally in a lot of plants, you see them right on the surface. And so it took me a little while to figure this out. This is a long ongoing project. Hey, Ruth. Anyway, so one of the things that's really great about the Princeton Church is that we have like a quiet body of fresh water, and a lot of plants just fall in. And because plants self-prune, we can kind of catch things as they go along through the season. So here's a flower. This is what we would call an oblique section through a flower. So here is the outer petals and the base of the flower. In the middle, we have the ovary. And this little ball in the middle is actually a future seed. We have a continuous series. So when I first looked at this, when I started my PhD, it was upsetting. There were so many types of fruits, but I started to realize as I looked through more material and I could connect things, that what I was seeing was actually different developmental stages preserved in the rocks. So here's a later flower. You can kind of see the remnants of these anthers here. So this is a stamen, it makes pollen, and this is where the ovule develops. And then later, you'll see these immature fruits where we have two integuments, a typical angiosperm trait, and a little embryo developing here in the middle. And it goes on like that. And so, and we have these, I love these stone cells. These make me happy. They're so beautiful. So we have these stone cells. They're in the stems, they're in the base of the leaves, they're in the inflorescence axes, and they're in the fruit. And so, and also all these little dark spots, this is a thick peel, so a deeper etch, more acid. They fluoresce, they just glow if you put UV light on them. And we have transitions between immature fruits like this, young flowers to fruits, and then later stages, and then even later stages. So here, this layer here, this is a really like spectacular and beautiful layer that develops over time. And when you see it in like face view or paradermal section again, it looks like stars. And so I realized that we didn't have many kinds of little fruits. We had one kind of fruit. And that helped me understand the flowers later. So I'm kind of doing this backwards. Paleobotanists like to start with the good stuff and flowers, but I'm going to go backwards. So as the fruits mature, so this layer here, it actually gets longer and becomes more star-shaped, like very much like epidermal cells. So you can see that here in the bottom right. So it kind of like gelled everything together. And because I have a lot of insecurity, it's yet to be published. But I have done the flowers, or the fruits, I mean, the fruit developmental series. So here are the flowers. So we have different stages again. We have things that are obviously seasonal in bud. So when you're in a bud, you have these, and we call them bracts, and they enclose all the structures that are inside. And the structures inside are small, and they're almost like preformed. And then after the season goes on, they open up and expand. So the shape might change more or less, but you can kind of figure out what's going on with a plant if you see a bud. So this one here on the left is a little later, a little more mature. And the one on the right, you can see the little bit of the ovary in the middle. And we have some textbook Lauraceae things happening besides the already established idioblasts, inflorescences, or sorry, trichomes, and what's the third one? The stone cells.
Stone cells.
Thank you, Ruth. Okay, so you can see, so this is a really cool thing about Laurales, which is the order that this family is in, is that it's the only family that puts its stamens outward. So most flowers, they have the ovary in the middle, and then the pollen producing structures, the anthers. The stamens are the full structure, but the anthers are the sporangial structures. They dump their pollen inside towards the middle of the flower. But Laurales were smart. They evolved early and they put some out. And so already we know we're in Laurales just by this one picture. Okay, so I'm trying to go fast because I know we're a little behind. So see this circle, this ovular structure? This is because a textbook Lauraceae, the only family that does this, they open their anthers to release pollen by a flap, like like a doggy door, and it's really unique. And so we know we have Lauraceae here. This is an immature anther with pollen developing. And then another textbook Lauraceous structure or trait is that the inner whorls, or the innermost parts of the flower, they have these little nubbins we call staminodes, and they tend to be secretory and produce sugars and nectar, so they attract pollinators. So already we got Lauraceae, no problem. But look, Princeton chert is so amazing. We have immature flowers all the way to mature fruits. So we have these young fruits here. You can see that the anthers have fallen off. The base of the flower is still there. We have later flowers, still the base of the flower there. And on. We have two integuments. We have meiosis occurring here to make megagametophite, or what we call an embryo sac and angiosperms, and all the way to mature fruits. And so you can kind of think of these as like baby avocados, or if you're from the west of the US, spice bush berries. So one of the things that made this project complicated for me is that the flowers are very confusing. We have incomplete preservation. You can kind of see this here, right? But Lauraceae is canonically trimerous, which means they have 3 parts to each whorl. And a whorl is, a level of organs. So we have outer petals or tepals, higher petals or tepals, anthers, and so on. And in the middle, we have a gynoecium. And there are 2 confounding factors. One is that in complete preservation, this looks like it has fewer parts, doesn't it? And then I also realized that there was something more complicated going on. So we have different stages of preservation, or sorry, maturity. Hi, yes. And then there's in inflorescence. And so you have to realize that in this family, some species have male and female plants. They'll have male and female flowers. And so I spent a lot of time wondering if that's what we had. But after I spent a bit more time using like 3D reconstruction software and looking at these preparations a lot, I realized what we had was a very interesting plant. We have the canonical 3x3 series at the tips of these inflorescences. So here at the apex or at the tips of these branches. And on the side, we have dimerous or two-parted whorls. And two-parted whorls is common, not that common, but it's common in the family for spicebush and bay leaf, for example, but only in the males. So it's been like a bit of a journey. So the most of the flowers you can see well, they have this canonical 3-part by 3-part. These little nubbins here in the drawing are the staminodes, the glandular protrusions. And that's very typical of the innermost whorl. And sometimes there's a 4th whorl and they're like undeveloped. They basically look like undeveloped stamens, but they're also usually glandular. But this one doesn't have that. So you can kind of see here, I've got in blue, the innermost whorl, purple next, orange, and then 3 parts in green, 3 parts in blue, and then the 2 gray bracts, 1 bract for the flower and 1 bract for that axis that produces at least 2 other flowers with it. So once you realize you've got bracts, all the numbers make sense. And there's some very like classic dimerous flowers that look just like bay leaf. I think there's a picture in the corner. Oh, well. And so what I figured out is that this flower, just by itself, just looking at flowers, doesn't fit any modern genus. But things like spicebush, they have a lot of variation. And so it fits more ecologically with spicebush. Spicebush grows in swampy areas. It grows with redwoods. It grows with decadon or swamp willow. It grows with sororus. But you can see like cinnamomum and litzia, they have dimerous flowers, and Laurus has dimerous flowers. But normally these are on separate plants. So this is a special plant that kind of combines traits from different genera as you expect in fossils. So here's kind of the floral diagram. At the apex of the main branch, you have this trimerous flower. And then the side branches at the apex, you have the trimerous flower. But the side, its buddies on the side, they're dimerous. So this is actually not unknown in modern Lauraceae. So that's fun. All right, so with the fruit traits, we have variation from that cup from the base of the flower coming all the way up at the outside at the end, just like kind of like an apple. And then you get everything in between too, like nothing to halfway. And you get 2 sporangia to make the flaps, to make pollen, or you can have 4. And in our species, we have 2. So this is a picture of, I believe, umbularia, which is a California native with 2. And it doesn't quite fit with what we have. And oh, sorry, on the left, you can see 4. I think there are 2 there. The left's probably Persea, right is Persea, sorry. And the problem with this family is that when you find fossils with plants, flowers are like, you know, they're the jam, because we know most of our descriptions and our ideas of what a plant group is are usually based on reproductive traits. But Lauraceae is a strange family where if you find isolated flowers, all you know is they're Lauraceae. You can have a combination of traits that are found across the family. So if you're interested in incorporating modern and fossil data together, we don't have a lot of information. We just know we have Lauraceae. So this plant will be amazing for anyone who cares about the evolution of this group. If you're interested in Lauraceae evolution, you have lots of fossils. We have about, I don't know, on the order of, what do you think, Ruth, 20 flowers. But all we can say is that they're Lauraceae. We can't put them in a clade. We can't put them in a group, right? So, so having all these vegetative and like reproductive and developmental traits is like really interesting. And I'm really looking forward to our next steps. So this is Lindera, the spice bush. It grows in the eastern U.S. It climbs up into the U.S. Canada, just like in the I don't know, that little wedge between the Great Lakes. But it's for the most part a warm temperate species. And the thing about temperate species is they have bracts because they have buds, right? So just by the inflorescences alone, I think we can infer that even though the Okanagan Highlands is probably warmer than now in the Eocene, it was probably seasonal. We have a lot of information just from a few little traits, but you can see, sorry, this is Lindera. It has two sporangia and it's dioecious, so it has male and female plants. And this is a male plant, and you can see these extra like triangular extensions, and these are those staminodes. So it kind of looks, and Lindera can be dimerous to like 16-merous, right? So just in that one genus, all the variation that we see in our fossil fits in that genus, and ecologically it fits. So that's why I framed it that way in the abstract. And that's it. A little overtime, but